time we stop here. So we discussed the two types of receptors. So we may have internal receptors that are going to be located inside the cell, and then we have cell surface receptors. Those are going to be protein receptors that kind of span through the cell membrane. So they're on the surface, on the cell surface. Okay. So we're going to take a closer look at each type of receptors. So this is the graph, the figure that demonstrates how internal receptors work. Okay, so let's see. This is the cell membrane, and this is going to be the extracellular space. So this is the exterior, and then this is the interior intracellular space. So this is inside the cell. All right, now you are going to have signaling molecules, which are, if you remember, also known as ligands. Okay, so internal receptors combine to ligands that are small and hydrophobic, right? Small, hydrophobic, so that they can cross the cell membrane very easily. Once they're inside the cell, they're going to look for their intracellular receptors, and they're going to bind to the receptors. And then this complex is going to move into the nucleus, okay? And usually this binding is going to cause a conformational change. So there's going to be an area where the receptor can bind to DNA. Okay, so a lot of these intracellular receptors are transcription factors, meaning they can affect the transcription from DNA to mRNA. So that's transcription, right? From DNA to mRNA. And then this is going to modify the process, the gene expression process, where DNA directs the synthesis of proteins. Okay, so once the receptor binds to DNA, like I said, it's going to increase or decrease transcription of a particular protein. So it's going to increase or decrease the production of proteins. Okay, and then we mentioned this before, that proteins are actually the workers that do all the jobs in the cell. So once you modify the type or the amount of proteins inside the cell, that's going to, kind of cause a change in the cellular activities. Okay, so that's gonna, so the end result of this transduction pathway is a cellular response, and then that's because you change the proteins. Okay, all right, so that's internal receptors, and then cell surface receptors. So again, I keep saying this because this is very important. They bind to water-soluble polar molecules, right? Water-soluble polar molecules, and that's because those molecules cannot cross the cell membrane very easily, so they can only bind to something on the cell surface. Okay, this part, the three domains, you don't have to memorize that, just kind of be familiar with it. So there are three kind of main components. All right, so this is the cell membrane, the phospholipid bilayer. Oops. Okay, all right, now let's say that you have a cell surface receptor right here. Okay, so this part that kind of facing the exterior of the cell in the extracellular space, this is going to be the external ligand binding domain. Okay, so ligand is going to bind to this area right here. Okay? And then from here to here, this is going to be the part that's inside of the membrane that spans through the membrane. So this is the hydrophobic membrane spanning region. Okay, so this area of the protein receptor is very hydrophobic because it has to interact or come into contact with the fatty acid tails of the phospholipid. Okay, so this area has to be very hydrophobic. And then the last area is this intracellular domain that's on the interior of the cell. Okay, all right, so those are the three components. And then the next one, the three main types of cell receptors, this is very important. Okay, so make sure you know how each one works. All right, first we're gonna look at the ion channel linked receptors. Okay, so this is how it works. You will see that for each type of a receptor, I will have a figure that can demonstrate how it works, how the signaling transduction pathway works. So you have to understand the process for each receptor. On the test, I will have very similar figures. So you don't have to memorize all the details. You just need to know how to read the figure and then figure out how things work. Okay, so what happens first, and then that's going to activate the next step, and then the next step. Okay. All right. This is ion channel linked receptors. So based on the name that you know, this receptor is kind of basically an ion channel. Okay. This is going to be the membrane, the plasma membrane, and then these green parts are going to be the cell surface receptor. Okay, so you are going to have a signaling molecule, the ligand. Okay, so the ligand can't pass through the membrane, right? So it's going to stay in the extracellular space. All right. Now, when it gets to the cell, it's going to bind to this ion channel linked receptor. And then this is going to cause a conformational change in the receptor structure because it's a protein, right? So it's going to cause a structural change in the protein. And that's going to open this ion channel, okay? So originally, the receptor stays or remains closed. But once the ligand binds to it, it's going to open. And once it opens up, the ions, these purple dots, they can diffuse, right, through this receptor, and then they get into the inside of the cell. Okay. So a couple things. We know ions carry charge, right? Whether it's negative or positive, but they're definitely polar, so they can't cross the cell membrane. So they have to, pass through a protein channel to kind of enter or leave the cell. Okay. That's the first thing. And then the second thing is that you need to know what process by what process that the ions can get into the cell. So once the channel opens, the ions are going to diffuse into the cell, right? So if you look at this figure, you see these purple dots. So you definitely have a higher concentration on the exterior and then lower concentration on the interior, right? So once the gate is open, then the ions are going to follow the concentration gradient. So they're going to move from a high to a lower concentration area. So they will move into the cell. Now, if it's the opposite, then the ions can diffuse out of the cell. Okay. All right. So again, you got to have the ligand, and then ligand binds to the receptor, and that opens up the ion channel, and then the ions can kind of diffuse in and out of the cell, right, depending on the concentration gradient. Okay.